Thanks. So it's fun to give a talk at Toronto. I rarely do. And so I'm going to speak a little bit about the philosophy at the beginning, because this is really more a philosophical question about how much care should we take about our science and why do we do science in the first place. And I love this quote by Giraudoux. So just as a background for those who don't follow the scene, our organization is a public-private partnership. It's a charity based in London, England. It was founded in 2003 when I started it. Now it has labs in 6 countries, about 300 scientists. And we're funded by 9 different pharma, the Wellcome Trust, Gates Foundation, Canada, Ontario Generous Funders, Brazil government, philanthropy. And in an interesting, sort of an unusual take, we never file patents on any of the results we have, despite the fact that we've had maybe $200 million in funding from pharma. And that's because the quality of the science and the availability of the science is an important aspect it's important to industry. More important than owning it is to be able to reuse it and that it's reproducible. We've had electronic lab notebooks since 2004, which I think is an understated but absolutely critical part of reproducible science. Rather than scribbles on a notebook that's in somebody's shelves that never sees the light of day, every single one of our experiments, I can tell you exactly what every postdoc did in June the 7th, 2006. The scientific impact of an organization like this is great. We publish obviously a lot of papers, 2.5 papers a week, and I don't know, we have 60 Nature papers or something. But we've done 13% of all the human protein structures that have ever been done, and about 25 clinical trials have been launched from the science we've done. So the organization is a big partnership to do science for the public good. So why? Why do this as opposed to running a lab, which I used to do? So akin to what was spoken about before, every year, you spend about $250 billion around the world on biomedical research. But for most diseases, we don't even understand how they work. Parkinson lived in 1808. That's when he described Parkinson's disease. We know a few genes. We absolutely do not know what causes Parkinson's, nor even a clue how to cure it or treat it well. Alzheimer's lived in 1909 or something. That's when he first described Alzheimer's. We still don't even know how that works. I mean, we're fundamentally doing a really shitty job of trying to explain science. And we all, the fallback argument, science, biology's hard, just give us more money. Well, I honestly think we should revisit that sort of simplistic statement. And even worse, when we do make medicines, they're not affordable for most people on the planet, which is really a tragedy. And so this doesn't make sense, right? You've got a fundamental disconnect, $250 billion a year. We don't know what diseases we have. When we make medicines, they're expensive. That's a sort of, you say, cognitive dissonance. And so if you look, though, at us, how we behave as scientists, now the young people, it's not your fault. It's old fart's fault like mine, okay? And so an interesting way of mapping how science is done across the planet has been afforded by the human genome. In no other region of science can you make a box around your scientific endeavor. But there are 20,000 approximately genes in each of us, and that enables us to say, well, let's look at how science maps on those genes, because each gene has a name. You can go into the PubMed and say how many papers are on this one, this one, this one, this one. So we did it for protein kinases prior to the sequencing of the human genome, right? And so here's the protein kinases. Many of these hadn't even been discovered yet, so clearly no papers on them. And you get this power law distribution, which no matter what, you get a power law distribution. So don't focus on the shape, but focus on that most of the papers are on a small subset of genes. Could make sense. Now, the whole point of sequencing the human genome was to reveal the mystery and the totality of humans. And remember Blair and Clinton shaking hands. This is going to transform biology. So I decided, well, let's have a look at this graph, same order of genes in 2012, and we've done it since that. And how did we respond to the sequencing, to this unknown that we were supposed to walk out to? Unfortunately, most of the science done today is on the same genes that we found exciting in 1999 and before. And this tells us about our resistance to change. It tells us about the culture of science. And it's the culture of science and the incentives of science that lead to the reproducibility issues we're having, right? We are rewarded for big publications. Try publishing a paper on this protein. Try getting a grant on this one. You won't get it, right? I want to study kinase 52 or 512 because we don't know anything about it and I think it's cool. Triage, right? I want to study this kinase because the mouse, when we mutate it, does this hop and the thing, and it's a new discovery for the well-known kinase, and now we can link it to this, link it to, that's a real word I hate, right? It's linked to this, it's linked to that, because it implies some functional interaction what's not there. And that gets us grants, because as reviewers, we think, well, that's an elegant thought, right? So what happens is, over the course of time, you set this plowing off of research on what we've always worked on. And you know, you can tell a funny narrative like I just did, but these are real data. You can't ignore them. That's BRAF, so when they get exciting, everyone piles on, right? So I asked Tony Posson once, why is BRAF way down here? He goes, well, Al, because there's ARAF. And clearly A is more important than B. And if you look at it numerically, it's almost always number 1 has way more papers than 2, than 3, than 4, than 5. It's completely idiotic, right? So remember, this is us. This is how we behave, okay? And what's even more distressing, I think, for the global use of funds is, well, first of all, you have to have a hypothesis when you're looking at this stuff. So my hypothesis was Canadians are smarter than everyone, and we wouldn't be doing this, right? And so we said, let's look at only the papers published by Canadians. Take all the subset out that says kinase plus Canada. Now, this is really distressing, right? And because what it tells us is that the culture of science, obviously, is global. We read the same literature. We basically have the same ideas. But our grant funding is parochial and national. We fund science not to better humanity, but for the economic interests of our country, to train people in our countries. And as a result, the same grants get funded over and over again. So Canada, every scientist in Canada could get hit by a bus tomorrow. It wouldn't affect the planet that much, because approximately everything's being done elsewhere. So you have a $250 billion budget, but it's sliced into little parochial packages, none of which is impactful that much. And so now you ask yourself, why aren't we explaining diseases? I think this is part of the answer. And the other answer is the care we take on individual experiments. So when I'm asked, why did you, right after the genome sequence, this had been happening in yeast and in bacteria before, so I didn't think about quantifying or visualizing it this way, but intuitively we knew this even when I started the SGC in the early 2000s, and this was bottom line the reason why we did it. So how can you fix it, right? Because problem statements are easy, I said before, fixing's the hard part. So how do we change this sort of mechanism? I can't change the culture of science, but maybe we can figure out a way. So one of the ways we thought about was maybe people are discouraged from studying the unknown because there's no reagents to them, and it's hard, right? So by the time you make all the reagents, your grant's over, right? So you can't afford to. So you see a big long list, you pick the gene, you can make the cell, you got the cells already, you got the antibodies, right? So maybe if we start to generate things to the right-hand side, it'll be better. So we basically launched over the last 15 years 3 significant operations to create reagents for protein structures, so structural information for the public domain, tool compounds, small molecule inhibitors of proteins for the public domain, and recently starting to think about antibodies for the public domain, right? And it sounds sort of boring, reagent generating, but anyone who's been in a technology development part of the lab, you have a new technology, it enables you to ask new questions. You have new reagents, you can ask new questions, and you can get the high-profile papers because you're the first to be able to ask that question. And so it is really kind of a neat way of approaching science. And of course, if you work only on the proteins that no one studied before, everyone is a fun mystery. So the generic strategy we've adopted since the beginning was, first we established quantitative criteria for the reagents we're going to be generating. Then we put in place an independent team of assessors to say whether we met those criteria before we put the reagent in the public domain. And then we make a commitment to share all the methods and reagents and everything without restriction on use so the world has access to it. So for protein structures, we did exactly that. We established quality criteria, and I was sitting up there finding a screen grab of it, right? So resolution criteria, all sorts of geeky criteria about the statistical data. And so we had this and that. If our structures did not meet these criteria, they weren't accepted. And we had a numerical objective for our board. We needed to do 350 structures in the first 3 years. Every single one had to have this. And we had an independent science committee saying, did we meet it or not, right? And so the bar was high. The incentives were aligned, right? If we didn't do it, we didn't get funded. So we needed to meet the quality criteria. Of course, we made the quality criteria because we passionately believed that it was important. And so we set up the scientific committee. We weren't on it. We reported to it every quarter. And we now put 2,000 crystal structures into the public domain. Every single one meets the quality criteria. And about 13% of all the available structures about human proteins were done by our groups. And interestingly, there was one pharmaceutical company whose corporate philosophy from the very top was, we never join consortia, it enables the competition, joined us. And why? He says, because we can repeat what you do. Now this was in 2005, we thought, wow, crap. I wanted it because we're so smart and you guys are brilliant, and it was sort of mundane, but obviously in the subsequent decade, it became realizing why this was important. They said, we can take one of your protocols, one of your clones, and we can do this crystal structure, and in 2 weeks, it's in our labs and we're doing science, right? If we take another thing from one of the academics, who knows if it's gonna work. And they made, I mean, these every time they join us, they pay $8 million, right? So it's not a small investment of their resources. So we then moved over in, let's say 2007, we said, we've got all these structures, why don't we start using them to make specific inhibitors of the protein? Because if you have a specific inhibitor and you give it, any one of you would know what to do to put it on your cells and ask the question, does this protein have a role in this process? Okay, so again, we established stringent quality criteria. So it had to bind to the target biochemically with 100 nanomolar or better affinity. Had to be 30-fold selective against related proteins. So for each protein, you have these off-target lists. So if it was a methyltransferase, it couldn't hit any other methyltransferase at 30-fold. And it had to have 1 micromolar proven on-target mechanism. Not you added to 1 micromolar in cells and the cells did something. You added it at 1 micromolar and you had proof that it went into the cell, hit your target, and the effect you saw was that. That's very hard to do, FYI, especially for proteins for which you don't know what they do, because it's all invention. How do we figure out that the inhibitor we have really hits the target we're thinking, and the effect is due to that? We again created an external committees to assess the quality criteria prior to the release of one of these into the public domain. We had numerical objectives. We would try to do 25 of these in 4 years. So far, we've released 50. We've distributed more than 10,000 samples of these. They're not, we file no patents. These are outstanding molecules, right? I don't know, you've spent time in farm. These are hard, they're about $2 million to make one of these. We give them away without restriction so that people can use them. Already 5,000 papers have been published using the tools that we've made. And these papers and the link between the protein and the disease spurred industry to launch now way more than 25 clinical trials, testing in the clinic the hypothesis that our compounds have generated in vitro. So it's a really home run experiment. But again, the process is the same, right? So do the reagents induce systems change? Do they cause people to work in new areas? So these are bromodomains. Bromodomains are a family of 55 domains in the human genome. They're 4-helix bundles, and they bind lysine when it's acetylated. So they're protein interaction modules that bring a complex to an acetyl-lysine group, and the lysine only when it's acetylated. And the compounds we developed sort of block the hole. And so the first one we released was 2010. So here's the trajectory of papers, and it continues like this if you say papers without the compounds. And that's when the compounds were released. So you can see that by creating research tools, particularly chemical tools, for unknown proteins, it really lowers their bar for scientists to study them. And there's a whole slew and these compounds all meet our quality criteria. And we're happy to say that of the 5 papers that the Reproducibility Initiative in Oncology tried to reproduce, they said, here are the top 50 cancer papers in the last X years. Ours was among the top 5. They reproduced the 2 out of 5, or maybe 1 out of 5 reproduced, and it was this one. Because I think you have very stringent quality criteria, and the reagent makes the experiment. So why is it important? So what happens in the opposite? Why did we think so hard and work so hard? It takes 18 months, 2 years to make one of these compounds. It's so tempting to put out a compound. Well, it's not quite there. Let's put it out, okay? And so what happens when you have a poor compound out there? So the most common search for a reagent nowadays is not by talking to your friend. It's Mr. Google. And so let's pretend you really thought you had an AMP kinase involved in your process and you wanted an inhibitor. So you look and you type in AMP kinase inhibitor and appears top hit. Oh my gosh, look at this. Tokris has one. High purity. Sounds pretty darn impressive. And then you get this, and oh my God, you're blown away. Look, it's got a chemical structure, which makes biologists like me go, ooh, that must be smart, right? It's got all this crap, potent and selective. Man, that sounds really good. Nanomoles, got no activity against zap and stick and, oh my gosh, it sounds great, right? Got molecular weight, solubility crap. It's really cool, right? And so you think, oh, I'm buying it. And then you can also find cell papers that use this thing, right? So you go, oh, Bob's your uncle. Okay, so these are internal screening data from Pfizer, where the dorsomorphin, which is supposed to be an AMP kinase inhibitor, actually inhibits 13 kinases more effectively than this one, right? All these. Oh, this is so common. I mean, you laugh, but you don't realize, right? So all these are hit. The actual in hand in Pfizer, it's 800 nanomolar. In the Tokris catalog, it's 109. And it's used over and over again. And now you start to look at these. We know in the community that these are bad molecules. How many are used, right? Well, it's known that this is non-selective, and yet 300 papers appeared in these 2 years. This is a PI kinase inhibitor that is the most dirty one. It hits about 20 different proteins, right? But at the time it was good, but now it's historical. But it's used in 1,000 papers, right? These are BCL2 inhibitors, 300 papers. These are thousands of papers. This PARP inhibitor even went to phase 3, doesn't even hit the enzyme. Blah, 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 blah. Okay, so we're giggling away, right? But what's super interesting is in all cases, there are great papers that show that these compounds suck. Right, so we imagine that the literature is self-correcting and all we need to do is to publish that it's a bad reagent and Bob's your uncle, the world will be a happy place with, you know, unicorns and rainbows and stuff. But that's not really what happens. So this is, again, a super interesting cultural way, right? If it's published in Nature, even though there's this real like cell death and differentiation paper that says it sucks, well, it got in Nature and the reviewers are going to look for, you know, a nice reference. All you can do, reference Nature. Oh, it must be good, right? And so we giggle about the reproducibility thing. We think, well, that's somebody else. It's not me, right? But indeed, it's all of us because, to be fair, how are you guys going to be able to read a paper on this compound and know all the pharmacology and know really what the off-targets are, know how to read it? It's really hard. So it's much simpler to see somebody else reference it and told you it was good and say, good. You know, I trust that. And so how do you do it? The literature does not self-correct. So we tried to figure out a way, and we co-funded with a whole bunch of other organizations, Wellcome and the Broad and ICR and us. We said we need experts to get expert opinion on this. So we launched this chemical probes portal about a year ago, and the idea is to give people advice. You type in this probe, you say, should I use it? And we'll tell you yes or no, or if in mice or not in mice, right? We have about 170 people from industry and academia signed up. They painstakingly go through the compound and tell you why you should or should not use it. You have rating systems. It sort of tells you and explains whether you can use it in mice, you can use it or not. Is it brain penetrant? Is it not? So these are really hard resources because this is, you know, saving somebody money is not as good a business plan as making money, right? And so it's incredibly difficult for these resources. So, you know, we're continuing to fund it. Our charity continues to fund it, and it's looking for money all the time. But the SAB members now are 160, and they devote so much time. These are scientists throughout the pharmaceutical industry, real experts. The users have gone up dramatically, as have the page views. The number of probes that we have in the site are the number of actual molecules that we comment on has been going up tremendously, and this is all due to the work of Amy Donner. Amy used to be, she used to be at the SGC, but she's also was an editor at Nature Chemical Biology, really knows her stuff. Those are the supporters, and I think this initiative, I mean, I think we created it to address a market failure, which is the literature does not self-correct, and as well, scientists don't, it's a really hard to be able to know a lot about pharmacology and medicinal chemistry if you're not trained that way, but the users are biologists. So this is a way we hope to be able to take our collective expertise in the drug discovery sector and allow people to have access to it. And so the plan for antibodies is really kind of exactly the same. It's a copy-paste. It's establish quantitative methods, create recombinant reagents, and make them available. And so the first question is quantitative. If you can't measure it, it's really hard to establish criteria. And so what we elected to do was to focus, I can't remember what's on my next slide. Yeah. What we elected to do was to think of the importance of quantifiable outputs, transparency, ease, and reproducibility. And so, as alluded to before, there's so many applications, so what do? I think currently the situation is paralyzed by there's so much to do, and that's why we've talked for 3 years and nothing's happened. So we decided to focus on something achievable, and something achievable was immunoprecipitation, only because once you immunoprecipitate from an extract your target antigen, if you solubilize everything that immunoprecipitated, you can trypsinize and inject on the mass spec in an unbiased way. You could say, is my antigen there? Tick, right? My antibody recognizes the native antigen. It's not overexpressed. It's not tagged in Western blot crap. It's like, it's there, right? And you can also say, what else is there? And always there's somebody else there. This is just protein purification like we did, like a Kornberg training, you purify something, something else is gonna be there, right? When you do this IP Western stuff, it looks beautiful, but you're fooling yourself, right? Just like the compounds always hit something else. And so this is a very transparent way of saying, this is my antibody's performance. It brings down my antigen, and you can use semi-quantitative methods to say this is the amount of the antigen I bring down, and here are the contaminants, and there will be. And the ratio between your antigen and the contaminants tells you about the selectivity of the antibody. And you can make those data public. So it's a really interesting way to provide at least one sort of signpost of a quantifiable metric to compare antibodies. And so we did this for thousands of antibodies, okay? And so we pay attention to potency. At what concentration do you add your antibody? At what concentration of extract? And so the method was, we ruled out all of these because at the time CRISPR wasn't there, so you couldn't properly make controls. We established a standard operating procedure for doing immunoprecipitations. This is just to compare the quality of the antibodies, right? We restricted ourselves to a single antibody scaffold, so we didn't have that variability. We had a single method to purify. We had a standard operating procedure, as it were. And then what we did is once we developed the method, we shared it with 5 other labs. They'd never done it before. We gave them 20 antibodies, blinded. We doped it with 4 that we know didn't work. We said, tell us what they do. Here's the method. And indeed, they spat back, that's an antibody to this protein, this is an antibody to that protein. This we couldn't really tell. Good. And so 5 different laboratories repeated. So it was all done by academics. It's absolutely not industry standard, but it tells it could become industry standard. So it worked, and that was the publication. And so now I'm thinking if I were to approach the antibody, what I would do. First of all, establish quality criteria for quality, and don't do it by consensus, because it's going to take forever. I've been sitting on these meetings for so long, And everybody's got their own little, because antibodies do so many things, everybody looks at their little slice and it's goddamn important to them, and they will talk for hours about it, and you think, my God, stop, right? And so it's fairly simple what you do. You know, the Western blot, get a CRISPR cell line, titrate the antibody side by side. You can probably do it, right? And dope in your antigen at the side so you get sensitivity. It's done, right? And it would take any logical person about 2 days to come up with the criteria, at least the starting point criteria that you could start operating tomorrow. But we're still talking, right? Years after, okay? Devote time and energy only to reagents that you can reuse, right? Because the polyclonals or even monoclonals drift, and by the time you do all the QA/QC, quality assurance, quality control, the reagents changed. So make sure you have it recombinant and sequenced so everyone has the availability. I think the field needs an independent assessor of quality. When Thermo, which is a collaborative of ours, puts their catalog images on there, I'd call bullshit, right? That is too nice a Western blot. There's no way that could possibly be done by a human being, right? And so I think there's a real issue. Yeah, I'm being a little facetious, right? Because they do aspire to high quality, but it's the pot, you know, it's a fox running the hen house or whatever. They create their own reagent, they do their own QC, and they show you the best picture possible, and then when you mess up, they blame you, right? So somehow we need a third-party QA/QC lab, like all sectors have, right? An independent reference laboratory to which antibodies get shipped, they go through a process, the data make public, then you know. I think that is a critical aspect of this. We're thinking about how to do that. And then, of course, don't put any restriction on use on the reagents, because that's just bad. So I think it's a fairly, if I was God, you know, this could happen tomorrow. I'm not, but we're going to try and push her hard for something like this, because this is really self-evident as to what would hard. The approaches of bringing together the funders and the journals, that's a perverse incentive. So that's a problem of perverse incentives, so I say. So, you know, ideally, journals don't accept the paper unless the antibody's been through a pipeline or a process. There is no such thing. So they say, come up with one. Funders don't like to do anything. They're the biggest wimps on the planet. So they just hand out money and say it's community's problem, right? And then they make big statements and editorials in Science, but actually don't do anything, right? Companies, they might want to do something, but there's no business incentive to. So if you sell 1,000 antibodies, actually only 100 make you money, right? And you don't know which 100. So you put a new antibody on the market. If you spent the money validating it, let's say it's probably $20,000, $10,000, $20,000, and nobody buys it, you just lost $10,000, $20,000, right? So you want to know, is somebody going to buy it before it's worth validating it? So they throw it out to you, and then they look at sales. Ooh, this is good. Now we'll validate it. And so they're doing it wrong, but you can't blame them, because the way that we do things, if there's no quality criteria, they can't do it. All businesses react to regulation, and that's true for the oil and gas industry, that's true for any industry, right? They won't change unless there's some sort of reason to, and there's no reason for them to change, right? And so they won't. So, and getting them all together, and they're all misaligned incentives, won't happen. So I think it's just up to someone to do something, and then my suspicion is the field will change. So that's what I hope, and I hope efforts like this can help, right, in terms of being able to provide more ammunition and more data to be able to convince the community to start things. But if it were up to me, I would just start something and plant the flag and say, this is what we're doing, and then let the world follow, because right now there is no leadership in the sort of whole sector, and understandably, because it's 4 different constituents and it's hard to find a leader. Okay, thanks very much.
So I'll open the floor up for questions. Thank you. Hey, thanks so much for the talk. So number 3, while it makes complete sense, I guess the only problem is there are like 3.7 million antibodies. and there's so many different combinations. You might think it's even in the billions with all the techniques available. So how do you do that in a way that's actually scalable or makes an impact?
I think you start maybe with restricting it to reagents that if you spend the time validating it, that it'll be good in perpetuity. So polyclonals, gone. Most monoclonals, gone. You're a fairly small set of reagents that you can start to sort of create a good knowledge base about. And I don't think, and then you'll start with one application, maybe in one small field at the time, right? Neuroscience or cancer. Figure out some way to parse it to make it a chunk that you can bite, right? And because what will happen is that's the typical response, and it's a good one. It's so complex and so big, you shouldn't start. And then 5 years from now, we'll be a little older, reproducibility crisis. Right? And so I think the way to do is to chew off something we can do and just begin and learn by doing.
Thank you.
Hi. Over here. Hi. What is your thought on the Science Exchange Initiative in California, which is supposedly supposed to be an independent reviewer and a lab that reproduces your science for you? What's your thought on that?
I think it's a waste of money. It sounds nice and everything, and it's really hard to imagine how, if they reproduce it, the science has moved on. If they don't, did they make a mistake? I think there's enough people reproducing it already if it's an important paper. And it's far better just to do it right the first time than to worry about all these sort of do-gooder police people going around trying to help everything. So I would be much focused. It's much more productive, as it were, focusing on creating, doing the hard work up front so that the subsequent work can be of high quality, as opposed to going around and cherry-picking papers that you might want to reproduce or not. So.
But could that lab be used to be the independent assessor of quality when you're just...
No, that's not the right kind of people.
Okay.
This reference laboratory is a bunch of mass specs that run exactly the same every week. And they take pride. You recruit people who like to dot I's and cross T's, and they will not sleep until the period's at the end of the sentence. And they're completely unbiased, and they don't have a, right? And if you had that, I think that'd be a tremendous contribution to society in this field of antibodies, to be fair.
So I had the same question for actually Dr. Friedman, and great talk, which relates to that as well. In order to do number 3, why is NSERC or CIHR or NIH and all of these other funding agencies just unable to regulate or unwilling to regulate any of these processes? Why aren't there higher standards that we can all meet? Because once you have a flag post, then okay, this is where I have to go. But it seems like we're lacking that right now.
Exactly. Well, it could, in one world, it could work like that, but how the funders have evolved over the last decades or maybe half century is that they're not active participants in the science. They don't see it as their role as police. They see it as their role to enable the community to do their jobs, and it's up to the community to develop their own standards and stuff. And you're right, maybe they needed to play a different role, but the kind of people that they recruit into funding organizations aren't the real kind of people to be making these very difficult science calls. And if you create number 3, it's going to have to be a for-profit entity because it's going to need to be sustainable. So you also need to put some management and financial skills in place on top of this, as well as technical skills. And so I just don't think that the skill sets and the interests and the raison d'etre of the funders match with this, right? And so, and as scientists, we all know what happens when the funders try to change stuff, as we've seen what happened in Canada. We don't like it. Right, so can you imagine the CIHR coming in and telling us how we're supposed to do our science? We'd freak out. At least the professors would.
Thank you.
Hello, I liked your talk. It's kind of the same question as what was just asked, but from a different perspective of why not trying to get industry to agree on some sort of funding for this independent assessor. for the companies which produce antibodies that we like, rather than the ones that just throw out the shit, could we not get them to agree, since they're already maybe involved in the SGC, to have some sort of funding into a laboratory or something like that that can validate their antibodies as a competitiveness standard?
It may happen like that. So this is you're coming from a very good place, but it's about tactics and strategy. If you have this meeting among them all up first, it can be really hard to get agreement because it's a concept, right? And they chat forever about who owns what, how it's going to operate. I think the path to the end is to create it, get it going, start to apply some pressure to them, and then they say, crap, I got to be part of this, or no one's going to buy my reagents. So I think where it's going to end is exactly what you say. It's going to be an initiative that's funded by industry. Maybe only you have to pay you will have to pay a service fee to sort of validate your antibody. But getting there, I don't think tactically getting these big organizations in the room at the same time to come up with a business, it's not their business to create one of these things there. So, but I think ultimately you're exactly right. That's what is going to happen.
Actually, can I just add? So we're actually trying to do that with this whole scoring thing that I described to you, and work with industry, and you know, thank you. And I am getting to the point of exactly that, which is, screw this, we're going to create the tool, we're going to create the scoring, and you know, they're either going to come in line or they're not, because for the very reasons that Alad was saying, that it's ultimately impossible, that they're not, their incentives are not perfectly aligned with ours, and you know, in the end, you know, we can say that we tried, but I think at the end of the day, we're going to just have to do it. And they're going to just have to.
They will.
Hey, so I really love these figures at the start of your talk about the papers that are being published about certain kinases and how it hasn't changed globally. So I'm really wondering if we're going to sort of get these companies to step in line and try to set in this quality assurance protocol for their reagents, aren't we kind of pushing these companies to go for the mostly researched genes or proteins? And how could we prevent that?
So the question was, are these companies, so there's two kinds of companies. There's the pharmaceutical industry, which we've convinced it's worth their while giving us a significant amount of money to study the that are understudied, because they may be future drug targets. The companies that sell reagents answer to different corporate objectives, sales, right? And they will simply track which reagents to which proteins buy the most. So they'll read cell, there's the newest, hottest protein, let's make an antibody to it. They won't QC it, they'll throw out to the community. If people start to buy it, then they'll get more figures. Interestingly, sales tracks with the number of pretty pictures in the catalog. That's what the marketing guys in the industry will tell you. So you just look at the more pictures and go, ooh, I'm buying that one. And that works. It really does. So their incentive is exactly as you say, to make reagents to the hottest proteins, which tend to be at that side, which again, self-perpetuates where we do our research, because there's no antibody to you do a gene expression study, you see 10 genes, you look for the one you recognize, you look for the one there are antibodies to, you know you can, I've been guilty myself, right? You know you can create a story, your job's to get your postdoc, a professor job, you think that's a cool story, that'll work, it works, you do it and you ignore what might be the most interesting ones, and it's a real pain, but it's how the system works.
More questions for Dr. Edwards?
I was interested in your chemical probes initiative. How do you decide which probes which inhibitors you are going?
Which targets?
Yeah.
Targets. So we decide. So how do we decide which targets we're going to make an inhibitor to? So first of all, there has to be nothing publicly available. Then we chose epigenetics as an area. This was in about 2008. We said there's 400 proteins in the human genome. About 20 are studied. There's at least one druggable member of the family. So there's 150 methyltransferases in the human genome. One was already drugged. We thought there's 140 not studied. Let's just go for it. We don't know what they do, but a tool compound is a good way to find out. And actually, from your background, as an aside, the idea for this came from GlaxoSmithKline and the nuclear receptor field, when in the mid-90s, they were cloning nuclear receptors, which are, these are nuclear receptors, they're not cell surface, right? They're inside the nucleus. They bind the ligand, hormone, testosterone, and once they bind the ligand, change shape, and there are transcription factors and induce transcription. Well, in the early 90s, about 6 were known. 6 of the ligands were known, vitamin D, estrogen, et cetera, but 42 were discovered. And the companies went, my God, these 6 are druggable, and there's drugs. There's 42 new ones. This is gold, man. But they said, we don't know what they do. How the hell are we going to figure out what they do? We don't have any biologists in-house, but we have chemists. So why don't we make inhibitors of them? We can set up cell-based transcription assays, screen for ligands, antagonists of the transcription activity, optimize them, and get really good compounds that inhibit nuclear receptor 51. We don't know what it does. And then they tried to figure out the biology internally and couldn't. Then they thought, what the hell? Let's let the community have the compound and let them play with it. And all of a sudden, the number of papers on each nuclear receptor go rocketing up. In every single case, the number 1 cited paper in the field is the chemical probe paper that first described GW blah, 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 blah. There are thousands of citations for each and every single one of them. They've been used to launch drug discovery programs. The new, I think it was an LXR ligand, was just approved last year based on the studies that were done. And so they're very powerful tools, as it were, to uncover biology. So we really copied a pharma strategy and just put it into the public domain. And the guy who did all that work's at the SGC now. Tim Wilson, do you know?
Like when you're developing the chemical inhibitors, you have a kind of quantitative measurement like KD and something. When you're translating it to antibody field and you're using the IPMS technique to identify them, how do you quantify them? Because the very principle of affinity-based purification, it would attract other proteins. So you have to set up a standard like our antibodies should be 10-fold higher than the other proteins.
That's
something like that.
So we did exactly that. And so first of all, we established a standard 2 mg per mil extract, 100 mg, 2 micrograms of antibody. They should be titered, right? So in a real experiment, they should be titered. And then we looked at the difference between the antigen in peptide counts, but that's not really fair because if it's a low abundance antigen, right, and the contaminants are in high abundance, it might not appear. Also, if it's a protein complex, you sometimes see 3 proteins appear at the same time. But what it does do is it plants sets a objective data for that antibody. And the next antibody that comes through the same process, if you see the spread between the antigen and the contaminants go up, you say, that's a better one, right? And now you also have the CRISPR cell line, so we didn't have them at the time. You'd obviously, the negative control is to do IPs from the specific knockout, and then you can tell which ones are contaminants, which ones are complexes. And it's just more data, because all this, it can be shared via a database, as you know, and so people can go in eyes wide open. There's no reagent on the planet that's specific. You just have to know. And now we don't even know.